All right, it is 10 o'clock. We are going to get started. There will still be some people pouring in. Welcome to Quantum Innovations. If you are new to us and you're joining this Zoom training today, we're so glad to have you. Exciting. This is the first time that we've done this from two locations. So Rich and I are here in the Oregon location, as well as a bunch of our staff in the Orange training room with Tim. Norm is in our Minnesota location. Hi, Norm. Welcome. We're glad to have you there. Norm will be leading the training today. And a little bit of housekeeping to make this training the most optimum for you. It will be best if you go to the upper right-hand corner of your Zoom screen and click on speaker view. That way you'll have the most of Norm and his presentation in your screen. If you have questions during the training, we welcome your questions. We ask that you put your questions in the chat box, the Zoom group chat. You can find the chat feature at the bottom center of your Zoom screen. And at the end of Norm's presentation, then we will answer as many of the questions that we have in the chat as possible. And with that, I am excited to turn this over to our resident expert and owner of Quantum Innovations, Norm Kester, live from our Minnesota location. If I can just figure out how to make him go. And, oh, I just, you unmuted it.
I got it, yeah.
Sorry, there you go. All right, there you go. It's all yours, Norm.
Okay, so thank you everybody for joining, and thank you for everybody on Facebook. Before I get started, I do want to mention that there are thunderstorms rolling into the area right now. I am in Minnesota, so when it rains, it pours, and so hopefully we don't lose power like we did last week. So with that caveat, I do want to thank a couple of people before I start the presentation. One is, of course, the MacGyver group. Without the MacGyver group, is my speaker working? You're muted. It just popped up with the speaker not working.
Yeah, it's a little bit choppy.
All right, it's a good time to catch that. How is it? Is it better now? Try a different speaker. All right. Sorry, everybody. How is that speaker? That speaker's better? Okay. So I did want to thank a few people before I get started in the presentation, and I'll share my screen in just a second for the presentation. One of them is the MacGyver group. Without the MacGyver group, I wouldn't have gotten involved in all the different things that I got involved with all the different hospitals and all the different people around the country. So thank you to those guys. The Asante Hospital Group, and then all the hospitals and area clinics and that sort of thing, getting involved with them and the people with the state of Oregon was very beneficial for me to learn, and for a lot of our people to learn about what's going on with coronavirus and COVID and the surge and all that sort of thing, and that was very beneficial. I also want to say thank you to the people at the University of Tennessee, both in the Knoxville location and in the Florida location. They were very instrumental for testing our UVC box, both at the BSL-3 lab and the BSL-2 lab. So we were able to test with the actual coronavirus and then test different bacteria and things like that. And lastly, Luxottica, who has been very helpful all along in this project. We were able to get UV box to them in Italy early on in their surge, and they were really instrumental in testing it against the actual coronavirus, and then a whole host of other things. So thank you to all of those guys. So I do want to start the presentation. Let me share my screen here. Can everybody see that? Oh, just give me a thumbs up. Okay, good. All right, I'm gonna hide you guys because it ends up being a little bit distracting. So for those of you who don't know who Quantum is, this is us, Quantum. We are creating solutions, and that's kind of our motto. That's one of the things that we feel like that we do really well at Quantum. And this is kind of a little tagline that we have when you hire us. You don't hire our products. You hire us. You hire the people. And our people are quite proud because we do a lot of very interesting things, but we do it with a heart towards service to our people, with a lot of honesty, integrity, and that's why we've been so successful at what we do at Quantum. So enough about us. So I want to jump right in, and I'm going to talk to you today. This is obviously a 3D model of the SARS-CoV-2, and the things I'm going to cover today are I want to get into the difference between antimicrobial, antibacterial, antifungal, and all of those groupings that really help to understand what those different terms mean and how they're applicable both to medicine, to surface treatments, to things like we're doing right now with coating applications to eyeglass lenses. What do all those things mean? What's the difference between cleaning, sterilizing, sanitizing, disinfecting, all those different terms? Where do they apply and what do they all mean, and where is their proper application? As well as kill and inactivate, which are terms that get thrown around a lot. And all the while as we go through these things, and also one other thing I want to talk about is the different acronyms that we've all been exposed to, and then what do they do? What do these people do, such as the FDA and the EPA and the CDC and the NIH and the WHO and all that sort of stuff. All the while, as we take this journey, I want to show you guys a whole bunch of new images of the coronavirus that have just come out. Some really killer electron microscope views of the coronavirus and people that have been infected. And a lot of the images that you're going to see today are from an actual patient that was on a cruise ship. And I got asked right away, when I was showing this to some people here in the local area, some of these images, did the person survive? I don't know the answer to that question, if they survived or not. As you're gonna see, there are some pretty, this person has a pretty gnarly infection. So we'll see that here as we go along. So jumping right in, the first thing I want to talk about is all these acronyms and what do they actually do? And what I like to do is break them up into two buckets. One of them is really one of the buckets is really talking around and really organizations around solving the problems, really understanding disease and outbreaks and all these sorts of things, whether they happen in the United States or around the world. And the other group is really the governing bodies that are there to serve to protect the population against us, like rushing a vaccine out to solve the virus without all of the research and necessary studies to really, before we roll that stuff out. So there's governing entities, and then there's really the ones that are on the forefront of really logging all that research, understanding all that research, and creating a really great library for us to go and dive into. And the two that were really, really helpful for me and for our team at Quantum is the CDC and the National Institute of Health. Those two organizations have such a vast library of information, and especially this was really important as we started to go down the road of how do we disinfect a whole bunch of N95 masks for hospitals like we did with Asante. And we just tapped into all the information that was already there with the CDC and the National Institute of Health for battling things like the Ebola virus and some of the other outbreaks that have happened in the past. And guess what they had to do? They had to reuse N95 masks, and they had to reuse gowns. And so they actually had already gone through all this. And so this is where UVC came to the forefront for us, was just diving into all that research. So the first one, and I'm not going to read a lot of what's here. You guys can read it for yourselves. But the CDC, what they do at the CDC, again, they have such a wealth of information around the research that's been done and the wins and losses and what's been beneficial and why, and what's been approved, but in different situations. Very, very beneficial. If you haven't been to their website, I really encourage it. If you do like information like this, they have just a wealth of it. Then the National Institute of Health, the NIH, and they are also one that has been really, really beneficial. And again, you can Google these, or if you do some research, if you Google into how do you inactivate a virus on an N95 mask, probably the NIH will pop up, or the CDC, because they have those documents, and they're constantly revising those documents as we learn more information. The last one that's in this bucket, whoops, I'm gonna jump to the regulatory side. So the FDA on the regulatory side, and really the where we're gonna parse between the FDA and the EPA is around critical and semi-critical. So the FDA is really going to govern a lot of things around critical and semi-critical devices. That can be drugs, that can be things that they do for the treatment of different diseases, surface treatments, that sort of stuff. So the FDA is going to take a real high level approach, some of those things that are in the high level. If you're talking about something that's used in an operating room, the FDA is probably going to govern that, because you're going to open somebody's body and do some surgery. So the FDA is probably involved in that. The EPA in general is going to regulate the disinfectants and sterilants used on surfaces, but not those used in the really critical environments. So you'll see those governed again by the FDA. And the last one is the World Health Organization. And the World Health Organization really is doing a lot of work in governing around vulnerable people. So you see, like when there's an outbreak in Africa or some parts of the world where there's a disease outbreak like the Ebola, the first people boots on the ground was the World Health Organization. They have a really, really broad organization that really is working hard to try and curb the impact to especially the most vulnerable, but they are working to, as you can see in their statement, keep the world safe. So it's a really cool organization, and they've got a really cool website if you want to jump in. I put a lot of this, one thing I should have mentioned in the very beginning, all of this information that I have in the presentation for you today, all of the links to all the data, including links to the World Health Organization, link to the CDC, link to the National Institute of Health, the EPA, the FDA, those links, I've compiled all that information for you. So you can actually go, if there's something that I used in a slide here like this directly from the WHO's website, I put a link to that in the information. So all of those data links are available to you if you want to access any of that information. So jumping right in to the nice sideways view of the electron microscope view of a large cluster of the coronavirus. And I just think technology is so cool. And as these images are starting to emerge, and you'll see some really cool images throughout this, I just think that I know this is a bad disease and it's causing a lot of really bad things, but I really am awed by the technology that's been brought to bear on this and a lot of other things. So we're getting exposed to images like this because of this coronavirus, which maybe we wouldn't have ordinarily, but this is a really cool view, and you'll see a bunch more like this as the presentation goes on. So one of the things that I wanted to talk about today as I kind of set the table for talking about inactivation and killing and disinfecting and sterilizing is why. Why are we concerned about this, and why should we be concerned about this outside of the coronavirus? So as I show in this graph, and this is a graph that you can get from the CDC or the World Health Organization. You can see this whole study around the impact globally for different diseases and stuff like that. We're considered, of course, high income in the United States. And what we can see is other infectious diseases are on the rise because of antibiotic resistant diseases, which I'll go into in a little bit. But also people aren't taking vaccines. And so you see that on the rise. And then So you see outbreaks of different types of things, and you'll see some of them here in just a few minutes. But also we have things like the flu and pneumonia, which are still really prominent. So that's a terrible term. The great thing about a technology like this and really educating the world population about all these different germs is that now for the first time, as we're learning how to deal with these things, the applications that we're learning for the coronavirus apply to all these other things. So we could really decimate the flu and pneumonia by practicing some of the things that we're doing right now. The common cold would have a very, very difficult time circulating in the population if we wore a mask or a face shield, right? Washing our hands on a regular basis. So that's why I kind of want to set the table for all these other things that we should, as we've learned to deal with the coronavirus, this has application across a wide variety of different things. So now these graphs are old, obviously. Well, they're new publications, but they haven't been updated with the effects of coronavirus. So you'll see that obviously this graph is going to be dramatically skewed when you introduce in here 2019 and 2020 with the coronavirus, because the impact has been such a huge impact globally. But what I do want to point out is, as we're looking at funguses, parasites, protozoan, bacteria, outbreaks of these, especially drug-resistant outbreaks, are on the rise, and they are really, really on the rise, and in a big way. And then we've got the zoonosis and these different types of outbreaks, and I'm sure that the coronavirus is going to be classified in here, but it could also be classified in here as it's a virus. But we'll see how they classify it. But the point that I want you to take from this graph is that all these things are on the rise. And I'll get into some of the statistics that you probably aren't aware of, as I wasn't aware of, and then how we can battle these things, just like we are with the coronavirus. So lastly, I wanted to show you this graph, which is something that I was just completely unaware of, is drug-resistant chlamydia is through the roof. Drug-resistant gonorrhea is through the roof. Things like whooping cough, mumps, and some of these other things that we think of that were eliminated due to vaccines. And then on the gonorrhea and chlamydia side, STDs that are now, there are some pretty dangerous ones because they're so drug resistant. These things are pretty pervasive right now through the population. So, and this is just in the US. So just so you know, this is not worldwide, this is just in the US. So we're starting to see that as it's coming forward, that these things are just getting greater and greater in numbers. So next up here is a better isolated image of a couple of coronavirus, and you're starting to see why they call it a coronavirus, and you can start to see the corona around the virus here, and we'll see an even better image of that here in just a minute. But again, with the electron microscope, it's just really incredible what they can do with it. So, so here now this has not been updated. I just saw today, there's a new updated chart of the influence of the coronavirus on this data. It is now number 3. So it just passed the coronavirus. Now it's just been going up and up and up this scale. So now it is number 3 on the list of deaths in the United States, and I'm not sure where it's going to end up globally, because globally you have things like malaria and flu-related illnesses and stuff like that. Globally, you still have such a huge impact. In the U.S., because of our access to medical care and drugs, it's lower the impact of some of those other things, but not so in the rest of the world. So we'll see how those things balance out. But right now, if this was updated, you would see number 3 would be coronavirus. So, but what I do want to call your attention to, unintentional injuries in that bucket is drug-resistant, antibiotic-resistant germs, and also pneumonia and flu are still a really, really dominant factor here. So in unintentional injuries is also automobile accidents and things like that. So that's not just a drug resistant bacteria and fungus and things like that. So at the tip of the iceberg, we've all been kind of really ingrained now in our minds and in our way of thinking that, hey, all this stuff is coming out of our mouths, it's on our body, right? We have bacteria, we have germs that live in our intestines, that live in our stomachs, that are on our skin. But we're also expressing and spreading germs, especially the unwanted ones, via our mouths. And we've really been come to really be indoctrinated into germs that are coming to us this way. But what I do want to talk about is some of the other germs that are out there. And I've got some pictures of some of the germs. And I've been researching so many germs, I can't remember all the names. I do remember this little spiky booger because it is gonorrhea. And this is a drug-resistant version of it. And it just looks dastardly, so I remember that one. But according to the CDC, 2.8 million infections in the US, 35,000 people died. So, so these are things that are prevalent in hospitals a lot of times when you go in to get surgery or something like that. You can be introduced to things like maybe you may be familiar with something like staph infections or some of the other things. Some terms you may another germ you may be familiar with, salmonella or E. coli or some of these other things that are out there. Seasonal flu, here's another one. 650,000 people died globally due to the flu last year. So in the US, 35.5 million people were infected, and another 35,000 people died in 2019 alone. So coronavirus, here's the coronavirus. Now this statistic obviously was from, I think it was last week. So 21.7 million people infected, 776,000 deaths globally. So that's a huge, huge number. And you can see that it passed the flu as far as total deaths globally. And then in the US, 5.42 million with 170,000 deaths. So that's why it's way up the scale as far as the total number of people that died. C. diff, here's another nasty looking one. I gave it its own slide because of the number of people that get infected and then also the number of people that die due to C. diff. That is a nasty looking booger right there too. So, so now here is an isolated image of a single coronavirus. And so now you can really see this electron microscope view of this coronavirus. You can really see that kind of corona around. It almost kind of looks like the sun, but it is again very cool with technology what they can do to show us these images. But this is a really kind of mind-blowing view of a single coronavirus. So, now. The question that gets asked a lot is, how do drugs become antibiotic resistant? Again, I'm not going to walk through and talk through every single thing that's on this slide. There are links to this if you do want to see some of this information. These are slides that are, these are PDFs that the CDC generates so that you can, I guess, hang them in your workplace. I don't know what they plan to do with these things, but there's a whole bunch of different poster versions of a lot of these different things. And this one I found on the CDC's website. But there are a number of different ways that a germ becomes antibiotic resistant. And one of the ways that we're probably most familiar with is, you go to the doctor prescribes you antibiotics. You may or may not actually need those antibiotics. You may have been prescribed those, like a general antibiotic for something that needed to be very specific. And then every time that germ gets introduced to an antibiotic, it learns from that and then builds defenses around that. Or you could be prescribed 10 days worth of antibiotics. You start feeling good at 7 days, so you stop taking the antibiotic. And so the remaining germs that are in your body learn to be have a defense to that antibiotic. There's a number of different ways, and you can see some of the different ones here. Any of them are bad. So the more that a germ becomes resistant, the more difficult it is for us to get rid of it. especially when it's inside the body. So here we are inside the body. Now this is a human cell that's been infected by the coronavirus. Pretty nasty image, but also really cool that somebody could produce something like this. All the little yellow dots are the coronavirus on the cells. So pretty gnarly. This person again was somebody from a cruise liner and was infected by the coronavirus. This was taken some time ago. So pretty startling image, but also pretty cool with what they did with the electron microscope here. So as we transition now we've learned a lot about all these different germs that exist and why we should really be thinking about practices that we should be practicing once we're through this coronavirus, which we will be. We'll have a vaccine and we will have learned, and some things will change forever for us. Hopefully one of those things that we've learned is how to properly deal with those germs so that we can attack some of these other germs. So my big hope is that we continue to practice some of these things and we institute some of the technologies, some of the capabilities. When flu season comes around, we take our vulnerable population and we protect them by wearing face shields and things like that. I don't know what the future looks like, but now that we have all this information, we should be able to behave differently. So as we transition into how do we deal with all these different things, here is a great slide that I found from Solo, and Solo is all over the place. They have a lot of different products, and some of them are in this same sphere that we're talking about. So a really nice graphic. So it goes all the way from cleaning, all the way through sterilizing. So sterilizing is at the top of the pyramid, cleaning is at the bottom of the pyramid. So a lot of times when we sanitize, disinfect, or sterilize, we still have to clean the object before we sanitize or disinfect, especially. Sterilizing usually is so aggressive, especially in the way of heat, that we may not have to clean. But a lot of times if we disinfect or sanitize, you have to clean first, because the thing that you're going to disinfect or sanitize with has to be in direct contact with the germ in order to work. The other thing that I wanted to hit on, and again, I'm not going to read all of these slides, you can read them yourselves, but one of the things I really wanted to hit on here is, just because sanitizing does kill a certain percentage of microorganisms, and you may have to clean first, like I said, with disinfecting, sanitizing, and sterilizing, there are dwell times. So you may have a very good disinfectant or a very good sanitizer, but if you don't leave that material on the object long enough, it doesn't have enough time to work. All of the sanitizers and sterilizers, especially surface ones, have that on, should be on the bottle. So if you sprayed a surface disinfectant down, it may work in 30 seconds. It may work in 10 minutes. You have to read the label and find out what type of product you have, and then make sure that that product is left on the surface for the amount of time required in order to kill those microorganisms, or you're just going to leave them. They're not going to be killed. Same with sanitizing. So again, it goes from cleaning at the bottom and then all the way up to sterilizing. And one thing that you should also know is that the more dangerous the germ, or the more critical the environment the germ exists in, the higher up the pyramid you go. And for a great case in point is, if you're in the hospital and you're about to have surgery, if you're in a surgery situation, just about everything in that room and going to be used on that person has been sterilized. And that's because that's considered a critical situation. And all those devices can be exposed to things that are quite dangerous. Or it can be that it's in a semi-critical environment, but the germ that can be living in that semi-critical environment is very difficult to kill, such as spores. Spores are very difficult to kill. So a lot of times you're going to see sterilization alongside spores because of the difficulty to kill a particular germ. As we continue on, I really love this graphic as well, because this really kind of lays out for us where we're going in the scale. So clean air, sanitize, disinfect, all the way up to sterilize. And then what is existing? Now they broke out viricide separately. Now it's important when you're starting to, when you're bridging a gap between drugs and surfaces or treatments or paints or any of these types of things. Some of these terms start to be blurred in their application. But if we think about it in a drug kind of aspect, you don't take an antibiotic for viruses, right? So we are going to take an antibiotic for bacterial infections in general, and maybe for fungus, but not for viruses. So, you know, you can start to see and parse some of these things out. But as we get into some of the other slides, when we're talking about the difference between antibacterial and antiviral, we'll talk about that a little bit more. But remember that this goes up in the scale from cleaner all the way up to sterilize. Sterilize being the most aggressive way of treating something. And there are ways of doing that, like vaporized hydrogen peroxide. Heat is like an autoclave or something like that is probably the most widely known version of a sterilizer. So those are some pretty basic methodologies there. So now here is an image of a very badly infected cells here. So the pink is the coronavirus, and it's all over the place. It's a very, very bad infection. This person's body is replicating the virus very quickly, and it's responding very aggressively to it. So again, a gnarly image, but the technology brought to bear is pretty incredible. So as we start talking about, I want to bridge now, I'm looking at right now, what's the difference between, if you hear the term antifungal, antimicrobial, antibacterial, antifungal, so an algicide, where do these terms cross over? Where do they mix up? How do we understand the difference between the two? Or sorry, there's 5 actually. So now in general, when we're in the sciences, just in general, we're not in pharmaceuticals, or we're not in surface treatments, or we're not in some of these other very particular sciences. If you were to say in those environments, is something antimicrobial, that would be, does it do something to inactivate or kill a microbe? And then for that, you might say yes. Now, when you're talking about something very specific, like a drug or a surface treatment, if you say antimicrobial, when you're talking about a surface treatment or a drug, it is going to be, is that a broad base? Is that going to encompass all of the above? So there's a difference there, and we'll see it here in just a second. But when you talk about something that is antibacterial, that does not mean it does anything for anything else. It can be antibacterial without being antiviral. It can be antiviral without being antibacterial. So we just have to understand, especially when we're talking about surface treatments and the way that we're dealing with different things. You can have a really great antibacterial that does not do anything for the coronavirus, for instance, or the flu virus. Or we can have something that really works well to inactivate viruses, but doesn't do very well for bacteria. One of the examples I like to use is our Fusion UV device here inactivates the coronavirus very quickly, right? So if we were just to put some virus in there in a petri dish, we would inactivate that virus in a short number of seconds, somewhere between 10 and 30 seconds, depending on the device. This one would do it very fast. But if you were to put C. diff in there, like is on this card, now that takes a lot more energy to inactivate C. diff or some of the other bugs that can live in medical environments, and the antibiotic resistant ones. This UVC box does inactivate those things, but it takes a lot more energy to do it. So one size maybe fits all as far as being able to inactivate it, but the amount of time that it takes, or maybe the amount that it takes, would be dramatically different between the different germs. So just want to caution everybody, if you hear us say antimicrobial. At quantum, we're going to say that it is treating all of the above. If we say it's antibacterial, that means that it is only affecting bacteria. If it's antiviral, it is only affecting virus. So we're dealing primarily with surface treatment, not something that goes inside the body. So now here is another pretty incredible image. And this is one, if you see in this zone right here, this is actually a cell wall that has been broken through by the virus as it has replicated itself in large numbers, and it has come through the cell wall and is going in hunt of other cells to infect. Pretty crazy image that they caught here as the virins were emerging from the human cell. Pretty crazy. Again, with disinfecting and sterilizing, the thing that I want to turn, we talked about the different methods before, but when we talk about when you're actually analyzing a bacteria or a microorganism that's living on an object, the testing methods to do that are there's a variety of them, and one of the most common ways is looking for the RNA of that material. Is it left? It's like our DNA, but it's the RNA of the germ, the microorganism. And do you have any of that left? Now, with disinfection or sanitizing, in some cases, you're going to inactivate the virus. Like when we're talking about coronavirus, the RNA is still present of that particular germ. It's just that that germ is considered inactivated, which is the common term. And that means that and I'll show you some graphs here in just a second, some graphics. That we've scrambled the RNA or the DNA of the microorganism, and we've broken its ability to replicate itself. So it's considered inactive. It is still present. It's just, it can't harm anybody any longer. Now, sterilizing, all evidence of that microorganism has been removed. So it is the nuclear option for germs. There's nothing left. You can find no evidence of that germ on the surface when you're doing those kinds of studies. So you need to do a different kind of test. So here is kind of a cool little graphic of the DNA or RNA of a microorganism and what we're doing to penetrate that. Now, one of the things that you're probably aware of, and I know that I wasn't really deeply aware of this until I probably had a casual awareness of UVC. I should have had a better one, but I did not. UVC is completely filtered out by the ozone. And so because our atmosphere filters out the UVC, we don't have a lot of knowledge or experience around UVC, but also a lot of things like microorganisms don't have a defense to UVC. So UVC is very short wavelength energy light and highly energetic light, and it penetrates deeply into the DNA and RNA of these, especially viruses, where you've got these enveloped viruses. It's very easy to penetrate them, and that's why something like UV, this fusion UV works so quickly, is that the light can really penetrate quickly into the microorganism and damage the DNA or RNA, and then they're unable to reproduce. And this is where the term inactivate comes in, because all we have to do is scramble the DNA or RNA. It can't reproduce and it can't harm you any longer. So that's what we're doing. Here is a nice graph of where that particular light hits us in the electromagnetic spectrum. So we're getting closer and closer to X-rays as we get into the different types of light. You see where visible light is there, what we're accustomed to seeing. Infrared is the longer wavelength, lower energy, used for like your remote control on your TV. Visible light, UVA, UVB, we're very familiar with because sunburns and tanning beds, and maybe in the optical industry, we're curing things with UV, but we don't have any UVC or really experience with UVC except for in the industries like water purification and air purification. So as everybody probably knows on this Zoom training, UVC has been used for many decades to treat water and also air. So something close to 90 years of experience. Maybe it's 100 years now. I haven't looked at the data when it was first started to be used, but very effective for killing a lot of microorganisms in water and water treatment plants, and also in air purification systems. So there's a lot of knowledge and experience out there in that area. The germicidal part, as we're applying it to the flu virus or the coronavirus or some of these other things, that's fairly new, but it has been being used in hospitals for probably close to 30 years. More and more experience with that as time goes on. Here is, again, another slide on how the impact of UVC is on all these different germs. And the takeaway that I want everybody to have here is that as we've learned about staph and gonorrhea and chlamydia and the flu and the cold and all these different germs in the coronavirus, they all can be inactivated with UVC. There is no microorganism that has a protection to UVC. There's no known microorganism that has a protection to UVC. So they can all be inactivated with UVC. It's just the amount of exposure and the amount of energy that it takes. 254 and up into the 265 nanometer range is optimal. One other thing that I wanted to mention is there is another wavelength of light. I've talked, if you have talked to me personally, you know that I'm very excited about the 222 nanometer, and there's also, I think, one at around 195. There's some wavelengths that don't produce ozone, which is harmful to people, but also can kill the coronavirus, flu, cold, all that. Any airborne virus can kill it in the air. As you cough, it kills it, and doesn't damage your skin and doesn't damage your eyes. So that's a very exciting wavelength, and I think that we're going to see more and more of that as time goes on. Hopefully, my fingers are crossed. So now here is another cell with a lot of the virins emerging from the cell. And I want to show you right now what it looks like, and I'll jump into a couple of slides real quick here. But I want to show you what it looks like when we're actually inactivating a virus in a Petri dish. So I think these next few slides are going to be pretty mind-blowing to everybody. Outside of the other ones that I hope you find these pretty mind-blowing, as I have, pretty incredible. This particular shot with all of these virins emerging from the human cell. So now here is a really cool Petri dish example. This was done, I don't remember, I think it was China. Again, all this information is available for you at the end of the presentation. We have all the data available. You can see this entire study. But what I want to point out is the mock, which is there is no virus present. So at this point, there is no coronavirus. At 0 seconds, the coronavirus is introduced to the cells that are in the Petri dish. Now, here's the really cool thing. This is 1 second exposure to UVC, and this is 60-second exposure to UVC. Now, this should be pretty startling to anybody to see this kind of evidence like this. We know, thanks again, like I said, to the University of Tennessee, Luxottica, the University of Padua in Italy, and some of these other ones, we know that it is working, and we know that we're killing the coronavirus and bacteria and all these other things. But to see this is pretty cool. It's pretty amazing to see this kind of an image. And again, thanks to Electron Microscope, we can really get in depth into this. So again, here's the same study. Here is the mock. Here is the introduction of the coronavirus. Now you can see what we want, obviously, is for it to look like this. And you can see after 1 second, look at the dramatic change between 0 seconds when the coronavirus starts to replicate and starts to have its way with the cells here. And then 1 second, 10 seconds, 20 seconds, 30 seconds, 60 seconds. It is just amazing to see this type of a view and to have this kind of science really come to the forefront and be substantiated. So this is a pretty incredible view, again, of the impact of UVC on these types of bugs. So in conclusion, I hope that you've learned the difference between all the different acronyms, the FDA, the CDC, the NIH, the WHO, all those different guys. And you've learned the difference between sanitizing, sterilizing, killing, inactivating, cleaning, all those different terminologies. And and some big takeaways from this around the impacts of UVC on a lot of these germs, and really the emergence and the awareness that we should all have around some of these drug-resistant germs and some of these other germs that are impacting our lives more and more, and they can all be treated with coronavirus. So if you do have any wants or needs to look at this particular unit, you can see them on our website, qtmi.net, or you can go to a great partner of ours, Santinelli, and they are selling this direct to doctors. So they've been a really great partner to us, and we appreciate those guys. So you can visit them. And I'm not sure if that's on Santinelli or Sios, which is a sister company of theirs. So with that, I'm going to take questions and conclude. So thank you all, and I appreciate you attending. So any questions for me out there in Zoom or Facebook Live land?
I don't have any here in the group chat. Krista, do you have any on Facebook?
Nothing on Facebook at this time.
What's that?
Nothing on Facebook at this time.
Okay. Was this informational? Did anybody get anything good information? See some really cool graphics?
Yes.
I was just blown away by some of these images myself. So hopefully. Yeah.
I want to add something, Norm, you probably know, but maybe not everybody. You are talking about UVC at 195, 222 nanometers.
Yes.
This is called the deep UVC, and I understand that some researchers are working to use as a light to illuminate the buildings and interiors and kills the viruses. It's in progress, but we hope will be soon available commercially.
Yes, I agree. Yeah, it is being used, and it is being primarily used in Japan and Korea is where the technology came out of. And it is being used there, and they've tested it in hospital environments and other environments, and it does work quite well, but it is still something that people feel like is a little bit unknown. So the technology is new, so it hasn't been rapidly adopted, and it's still quite expensive because of the cost to make these types of lights. So, but my hope is that we see this type of technology everywhere in all of our common places. Hopefully in the future it gets there.
All right, if anybody has a question live right now for Norm, we have a couple minutes left. You are welcome to unmute and ask your question. If not, I'm going to share my screen.
That just means I crushed it.
You did crush it, dude. It was amazing. This was so informational. Thank you so much, Norm. Always one of our crowning purposes here at Quantum, sharing knowledge, and we appreciate you doing that. The screen that I want to share with you folks for more information. Here you go. Maybe you can see it. I made a quick slide. So for more information, contact www.qtmi.net. Or you can send an email to info@qtmi.net, and we can get you those links that Norm mentioned. There are a lot of them. We're happy to share them. We love sharing knowledge with everybody out in the community and beyond. So thank you very much for joining us, everyone, and thank you, Norm.
Thank you. Ciao.